So in this final talk on cell signaling, I want to kind of review some of the fine points as well as go over an example in apoptosis, or programmed cell death. So again, I can't emphasize it enough, this idea that the interactome, this idea that these little machines are interacting with each other on a cellular level is so important. This is how things are done, how we move information around the cell, how the cell quote-unquote computes what it needs to do based on environmental signals. So the fact that we have all these different steps in interactions of interacting proteins really allows us to, one, amplify the signal and contribute to the specificity of that response. So again, when I keep showing you pictures like this, I can't show you enough of these, because you just need to be familiar with this idea that you've got information coming from many different proteins. All these things are floating around the cell, bumping into each other, and that's how they interact, and that's how they say you should go forward, you're activated, or I'm going to shut you down, you're inactivated. So this idea that also enzymes, these are enzymatic functions, can amplify the cell's response is important. So when we look at that signal transduction cascade, that phosphorylation cascade, for example, if you put some numbers on there, again, an active protein kinase 1 may turn on 10 protein kinase 2s. And each protein kinase 2 will in turn turn on 10 protein kinase 3s. And that's how we get like an order of magnitude in the number of enzymes playing a role in this pathway at each step. So the more steps you have, the more opportunity you have for amplification. This is something to really think about. Now, each step of the way also contributes to specificity. So different kinds of cells have different collections of proteins. We already talked about this idea that in each one of your cells, all your genes are present, but not all the genes are expressed in each cell. And the genes that are expressed in a nerve cell are very different than the genes that are expressed in a stomach cell. And that's why those cells are different, because proteins do stuff. And the proteins that are there direct a cell to be a nerve cell or a stomach cell. So at the same time, those cells are going to have different networks of proteins waiting to respond to certain extracellular signals through these cell signaling pathways. So these different proteins allow cells to detect and respond to different signals, but at the same time, you could have the same signal be detected by different cells and they respond differently. All right? Again, the branching and the complexity of those pathways are also going to allow cells to integrate more than one signal to make the quote-unquote right decision based on what is happening in the environment outside the cell. So if we look at this, you can have a very linear response where you have a ligand binding a receptor and the relay molecules lead just to a single cell response, right? Now you may have, example in cell B, a pathway branches leading to multiple responses. This may be early in development or wherever in development where you need multiple genes turned on to do something. Now you also may have crosstalk. So you may have, again, multiple receptors that act on one crucial enzyme to say, hey, I'm getting a signal from this receptor that says we should go forward, but I'm getting a stronger signal. There's more information outside the cell that says, not now, don't do this. So we're going to increase our inhibition of this enzyme, and hopefully we won't initiate that response. All right? So again, different receptor leads to a different response, and each cell is going to be different in that respect. Why? Because there's differential gene expression that is ultimately set up from development. Okay? Again, go back to that pathway. We see that little bar, it says shut it down. We see the arrowhead that says move forward. And so these are very complex systems, but this is why it's cool. This is how a cell works. Now, there are mechanisms to make this more efficient. We talk about scaffolding proteins. And what they're going to do is group different proteins that are involved in the same pathway together and activate them simultaneously. So a scaffolding protein may grab 3 different enzymes or interact with 3 different enzymes, and you get a single signaling response that will activate then all 3 at the same time, and you're moving forward. Now, what we also need to remember is that the termination of the signal is important. We kind of referred to this a little bit earlier, that when I have a ligand bound to its receptor, we have that protein now in its active form. but based on the affinity of that receptor for its ligand, will dictate how long that ligand is bound. And when the signaling molecule, when the ligand leaves the receptor, that receptor is going to revert to its inactive state. It's like, oh, it's gone. So this all comes back to chemistry, right? The concentration of the ligand and the affinity of the receptor for the ligand are all calculations that need to be taken into account to ultimately dictate the intensity of the signal from the concentration of the ligand, right? So how long is that ligand bound or not indicates the intensity of the signal. Now, what I'd like to do is talk a little bit about a very important cellular pathway. It's called apoptosis. Now, it can be pronounced either apoptosis or apoptosis, and apoptosis is generally the accepted term in America. It's from the Greek. It's a Greek word for death. It's programmed cell death or cell suicide. And this is very different from the idea that a cell just dies, it's lysed, and all its contents spill out. This is the idea that the cell, the body, the multicellular organism, has decided that this cell, because it's been invaded by a virus or it's damaged beyond repair, it needs to be basically recycled. So let's program a mechanism, a series of biochemical pathways to break it down, chop it up, package it in vesicles that then can be digested and reused. So apoptosis is very important because it prevents damaging enzymes from leaking out of a dying cell and damaging neighbor cells, right? So it's a controlled process of cell death, right? Now, a fantastic model organism that we really studied apoptosis in a lot is called Caenorhabditis elegans, or shortened to C. elegans. And this is a nematode, phylum Nematoda. It's a roundworm. And the cool thing about this is that it only has 1,000 cells. So scientists have actually studied this worm so well that they can follow it from a single zygote, which we'll talk about later, the single first cell that it starts, to all the cell divisions, each developmental pathway, each cell lineage, to the final organism, because it's relatively simple. So C. elegans has been a great model organism for the studying of apoptosis, where specific proteins that accelerate apoptosis override those that put on the brakes. And let's look at that example now. So this is kind of like a dead man switch. So what we have is active CED9, and this is located in the mitochondria. And so when CED9 is active, this enzyme is active, it actually inhibits CED4 activity. And CED4 and CED3, as long as they're inactive, everybody's happy. But what we're going to see is that when this particular cell gets the signal to say, hey, you're done or you need to go away, that is going to lead to signal transduction that inactivates CED9, which then allows CED4 and CED3 to become active, and that's going to lead ultimately to the apoptosis event. Right? So other proteases, we're going to start chopping up the proteins in the cell, we're going to chop up the DNA in the cell, we're going to start blebbing off portions of the cell so that it can be recycled. So caspases are the main proteases that carry out apoptosis. So that's just a term that I want you to be familiar with. It's a family of proteases, proteins that chop up other proteins. And what can trigger apoptosis? That specific signal, which is going to be important in development, which I'll show you in a second. If a cell is damaged in a way that its DNA is irreparable, right? So if I take too much DNA damage, like from radiation sickness, that cell is done. So we need to go through apoptosis and try to salvage as much as we can from it. Or if for some reason we're getting a lot of misfolding in the endoplasmic reticulum, that may initiate an apoptotic response. Something is so wrong with the cell that we're misfolding proteins to such a great extent, beyond some threshold, that it's like we just need to shut it down. All right. So apoptosis is important, especially during development. So, for example, we may see in the development of a limb, the development of the digits, it's easy to form kind of a paddle structure. But then in this case, what we can see is apoptotic genes are indicated by a fluorescent protein, and you can see that the digits are going to form as the cells between the ultimate digits go through apoptosis. So cell death isn't necessarily always a bad thing. It's very, very important in development, And so we need to have that mechanism in place, and it's very, very important to understand how cell signaling ultimately results in apoptosis.